Well, welcome everybody. We're very excited to get the show on the road here. And it's such a great night because we have more participants than we do staff. This is great. What a great kickoff event. Welcome to what is the first of what we hope to be a regular event, which is a research in progress update from the Teddy and Daisy studies that you all have been a participant in. And we welcome you to this evening and hope that we meet your expectations for the evening and give you plenty of opportunity to both learn about what your contribution has made a difference in and how you furthered science, as well as realize what a difference you make and grill our investigators. That's why they're here for you, so you can ask them all of your questions. They might ask me to say to be gentle, but they can take it. So I'm Judy Baxter, and I am going to be your MC for the evening and also provide a little of the lighter moments in the day to connect you back to all of the time that's been involved in these studies. So first, in addition to welcoming you, I want to do some introductions, and then we're going to go over the history and goals of Teddy and Daisy, because they happen to come together, findings and future plans, fun facts and behind the scenes at Teddy and Daisy, what being a participant in a long-term research study is all about, and questions and answer sessions with all of these, with the investigators. So, but first, for introductions, I'd like to first, our speakers are going to be Dr. Marian Revers, who is the principal investigator of both studies. You've seen his name on lots of stuff for sure, and at various and sundry events and ways. And Dr. Revers is also the clinical director for the Barbara Davis Center, and Dr. Suzanne Bennett-Johnson is our other speaker for this evening. Dr. Johnson is a key Teddy investigator and has been PI of several other related studies looking at type 1 diabetes. And she is also the 2012 president of the American Psychological Association. So it's our distinguished, and it's a great pleasure to have you here, Suzanne. And we're really lucky to and there's several of, these are our Denver investigators, and Georgiana Klingensmith is here, Georgiana, over here, and Dr. Ed Liu, right next door to her. So they'll be available for questions as well at the end, and we have Dr. Eisenbarth Norris and Dr. Steck, who aren't able to be with us tonight. So, and some of you may recognize these folks, but I want to give them a special call out. Rachel works with the data management team, and Kim is on the clinical team, but they're both our retention specialist coordinator team and the planners of this event. So, and our data management team, neither one of these could be here tonight, these lovely ladies. The lab team, some of you have seen the lab team in that as they're coming in and helping draw some blood. This is our current lab team. There's a few people whose pictures we didn't have up to date on the roster. So, and there's that clinic team. You all recognize them, don't you? I bet you recognize Michelle. Yeah, there she is in the back. And Tricia. And everybody else and many of the ones that were greeting you are from the clinic team here. And our administrative team, Adela, heads that team up, and she's here with us tonight. So that's who we are and who you may have seen around in various capacities. So who are you? You're the audience. You're the ones that are really the important ones of this whole puzzle called Teddy and Daisy. So we want to just get a feel for in the room who here participates as a Daisy family. If you're a Daisy family, stand up. All right. Let's hear it. And for you Teddy families who get to stand up next, you'll realize why such a big shout out needs to go to those Daisy families. You'll hear some of that as we come out. So who here is a Teddy family? You stand up. Great. And just on the off chance, I don't think there is, we tried to double check the RSVP. Is there anyone here who has a child in both studies? Yes! Oh, stand up! That's great. You're only one of 18 families that have, and you'll see how many kids are involved in these studies. So that's really very special for us. So thank you so much for being here tonight and for all of the incredible work that you do for us to make these studies possible. It really wouldn't happen and couldn't happen and be as sustained an effort if it weren't for your participation. So we hope this is a small way to give back so that you can connect up to what all that effort does. How long has this been going on? There's a song, isn't there? But anyway, okay. So I'm going to take you back even to the prehistory here, which is that this is how long NIH has been funding this work. Okay? So starting in 1983, Georgiana, we were there. You and I in this room were there for the start of the IDDM registry. And that lasted from 1983 to 1993, 10 years, and it was a registry study. It wasn't trying to observe and trying to figure out what was going on. It was just trying to get a handle on how many people had type 1 diabetes in Colorado. Okay? But NIH said, we need to do more. We need to do more with this to understand what's going on with type 1 diabetes. And Marian Revers came along, and there he gave this idea. I don't know exactly where it came, but the idea to screen and find kids at high genetic risk to try to understand the environmental triggers and consequences of what might be taking it down this path. And DAISY was an outgrowth of the IDDM registry. And a portion of the initial DAISY kids came from siblings who were of people who had been registered in the registry. Anyway, so DAISY's been going 19.5 years, 19 and a half years. And Teddy started 10 years ago, or nearly 10 years ago. And it was a collaborative, multinational, multi-center, so there's 6 centers around the world, and you'll see this in a minute. So total, we've got NIH has invested 29 study years. Huge investment, because we don't know the answers. And so in case you didn't notice, research is a process of delayed gratification. If you can't be in it for the long haul, you know, so we've been doing this a long time. So why DAISY and then Teddy? So this is really the slide that sort of tells it all, if you will, which is that the incidence, that's the number of new cases occurring in a year. The incidence of type 1 diabetes is increasing. It's doubling. Sorry, it should be doubling. It's increasing about 3 to 5% per year and doubling every 20 years, the number of. Okay? So the genetics of the population doesn't change this fast to account for this rising. It has to be what in the environment is triggering this kind of increase in incidence. And we hear a lot about type 2 diabetes and the obesity epidemic, but what's really important is that we understand that type 1 is increasing too. And this increase is what has really become the underlying justification initially for DAISY, and for studies in Finland, Sweden, Germany, Poland are who figures into this. And because alone we can't always figure everything out. Hold on. Oh, Bruce. Bruce, am I having Bipper difficulties? So I'm moving to mouse. Oops. So that justified DAISY, that increasing incidence, and there were studies going on, individual studies going on around the world, both in the US and in Finland and Germany, Sweden, as well as Georgia, Florida, and Washington and Colorado. But we were all doing things a little differently and had a little bit different focus. And there's a power in numbers, and there's a power from collaboration, that if we did it all the same, then, you think it's batteries? Okay. So together, NIH decided, and actually all of these institutes at NIH decided to chip in, if you will, into the TEDI collaboration. So many of these studies are still going on, like DAISY is, and that gives us the beginnings of what to do on TEDI. So we're always learning from DAISY, and TEDI picks up and does this across the world. So, and you'll see the difference in the numbers. And so, do we think we've got a Bipper? So the conceptual model guides both of these studies. You all were screened. Your babies were screened at the hospital, and we identified you as having some of the higher risk genes for type 1 diabetes. So it kind of put everybody on the same genetic playing field so that we could understand what happens to develop the autoimmune, the beginnings of the autoimmune pathway. But you'll notice here that not everybody who has genetic susceptibility goes on to this autoimmune pathway. So this big arrow here, this is about most people don't, even with a genetic background, okay? And so what we want to know, and why we ask you all these questions, is what might be initiating this process and what might be protecting people from going on to this process. All those questions, all those samples are about that stage, and then once you have the autoimmunity starting, not everybody goes on to get type 1 diabetes. What's promoting the clinical progression? What's protecting people from that clinical progression? When does it happen in the course of things? Is it that infection that you had 6 years ago that's related to now? We don't know. Well, we're going to find out. That's why we ask you all of those questions and take all of those samples in all of those different ways. So this is the model that has guided all of our questions, and for each study, both DAISY and TEDI follow the same model. We may ask the questions a little differently. We may collect the samples a little differently. But basically, we're asking and doing very much the same thing. TEDI's doing it in 6 sites, so we have to be particularly compulsive about how we're doing that so that we're all comparable across our sites. So who got here? This is what we did. We have 2 cohorts, what we call cohorts, in each of the studies. We have people who are what we call the general population cohort, and these are folks, like many of you, who do not have a family history of type 1 diabetes. And then there's a smaller cohort in both studies where either there's a sibling or a parent with type 1 diabetes. And so in DAISY screened a total of around 33,000 general population folks and enrolled 1,424. They didn't particularly screen They were just looking for folks with kids with a family history and enrolled 1,118 of them. Teddy, Colorado, screened in the general population 75,734 kids, and you'll find out some more facts around this screening, to enroll 1,232, and we systematically screened for our first-degree relatives, close to 1,000, and have 143 enrolled. Just to give you a flavor, Teddy Worldwide, across all of those 6 centers, screened 424,000 babies worldwide, just the way you were all screened. And the total cohort of Teddy comes in at is 86,077 kids are enrolled in Teddy worldwide. So that's the power of numbers that Teddy brings to this, that Daisy alone couldn't have that kind of power. But Daisy still keeps us informed and has been going so long, they have the power of longevity. Remember, Daisy's been going 19.5 years. So, but our goal here for these studies, where is this headed and why are we doing this? It's really to find the environmental triggers, virus, dietary factors, psychological stress. They're the factors that we're all asking you about, to think about how to develop ways of developing a vaccine, elimination diets, things that might be prevention strategies that would address those, and to make a case for public health screening and prevention. So, and if you can do what we did on that scale, perhaps there's an important way to identify people who are at high risk. So, Marian, I am going to turn it over to you. And Marian's going to give us some findings and the future goals here. Okay. There you go.
Thank you. I'd like to start off from thanking all of you for coming here today. It's, as Judy said, the first time we are having this kind of an elite meeting. It's not possible for us anymore to continue more entertaining meetings for a larger crowd at the Children's Museum. So we decided to shift towards those parents who want to learn more and give us direct feedback about the study protocol, how we do the job, and also science. What is on your minds in terms of questions? What are your observations from daily lives of your children in terms of what may be important and we may be missing out? I'd like to thank Judy for a wonderful introduction, and all of my colleagues, Suzanne, who came from Florida, and Georgiana and Ed, and those who are not here today, and all the staff who is working very, very hard every day on making those studies happen. So with this short introduction, I'll show you a few slides, but before I go there, I've been treating kids with type 1 diabetes for most of my adult life, and I guess I've helped several thousand of families like those that we see at the Barbara Davis Center. So just to make what I'm going to say more focused, I'd like to ask how many of you have a member of family, a close member of family with type 1 diabetes? It's a good proportion. I would say maybe 3/4. So you know, the most difficult day is day one, when we meet with the family of newly diagnosed child, and they have a lot of questions. One of the questions, or a series of questions, has to do with what they feel as sometimes as guilt, that they neglected something, that they, I'm sorry about that. Jenna, could you take this call? It's a kid on insulin pump, and I usually look at the care link. I call them back, okay, in about 2 hours. I'm sure it's not urgent. So, on day one, a lot of parents are asking us, did we miss a vaccination? Did we miss a screening? Did we give too much sugar or fat to our kid? And the answer is always, the stress. Two months ago, my kid was in a car accident. Could this be the cause of diabetes? And the answer to all of those questions is no. There's nothing you've done or nothing that you've omitted that could have changed the course of your child's life. This disease starts very early in life. So, like in this example, most of kids that we see are actually already in their puberty or just before puberty when we diagnose diabetes. But when you look back at the life of this child, the inflammation in the pancreas that we detect by measuring autoantibodies started in this kid around the age of 8. And it took about 5 years to get to the point that so many insulin-making cells in the pancreas were destroyed that this kid could not control his blood sugar anymore. But we don't even know for sure if the process started here. What we are measuring here are antibodies, which are like bullets that white blood cells shoot at insulin-making cells. And those are relatively simple tests, very reliable today. But there are speculations that there may be something else happening months or even years prior to the first signals that we can read. This is why in Daisy and Teddy, we are asking you to start early and come to the clinic often. So we can ask you all of those questions on a regular schedule and unfortunately hold your kid to get those blood samples or get nasal swabs or get rectal swabs. You know what I'm talking about, all of those samples that they may not necessarily like. But this way we are creating a grid of samples. So if some of kids participating in our studies develop autoantibodies at diabetes later on, we can go back and we have serial samples going back to age 3 months for Teddy kids and age 9 months for Daisy kids. Now, my friends Republicans tease me, how come Daisy is going on for 19 years and you haven't found the cause of diabetes? You are among those 47% who are completely addicted to government money. You sit in your office and just pretend that you are doing something. So maybe they have a point. I mean, we all would like to find the cause of diabetes many years ago. It just turned out to be pretty difficult. And when I finished medical school and published my first paper on epidemiology of type 1 diabetes, I thought I knew what was the cause. There was an epidemic of enteroviruses in the area where I was working, and it coincided with an epidemic of type 1 diabetes. So I thought, bingo, we know the cause. Now all we need is a vaccine against those viruses, and we'll get rid of type 1 diabetes. So 30 years later, and quite a few fewer, I'm sorry to tell you today that we still don't have a vaccine or special diet to prevent diabetes. And it's not from the lack of trying. We've tried in very well-designed, very well-funded clinical trials about 50 different things. In kids early on in this process, even before they developed any autoantibodies, at this stage, and even after diagnosis of diabetes. And to make a long story short, The only thing that works is chemo, immunosuppression. And the overwhelming opinion of doctors and parents is that immunosuppression that has to be kept for life is not the answer. It's not the answer to high blood sugars. That insulin is a much better approach, a much safer approach with much fewer long-term complications. So I probably spent too much time on the first slide. Let's accelerate a little bit. Starting from DAISY, I have to apologize. I'm going to try to tell you about 19 years of work and about 100 papers that we've published from DAISY in about 5 minutes. So it will be pretty concentrated and maybe challenging. Now, I also apologize for a glitch that I've just noticed. In the list of publications from DAISY, we stopped in 2009. And it's not true. We've published since 2009 about 15 papers a year. So we'll fix it next time. You'll get a complete update. And some of the papers that I'm going to summarize here have been published in the past 3 years. So the basis, the data that creates the foundation for all of those papers and reports are your kids and your participation in DAISY. We started, as Judy mentioned, from 2,500 kids, and over the past 19 years, about 180 of those have developed persistent inflammation in their pancreas, which is the first step to diabetes. And we can detect this inflammation by measuring autoantibodies. If they are present only once, or maybe just one antibody at kind of borderline level, we keep testing until we resolve the situation. And in about 20-25% of cases, it's a false signal, false alarm. The antibody turns out to be false positive, it's single, it goes away. But when we see 2 or more of those antibodies, it does not go away. We know that those kids will develop diabetes if they have 2 out of 4 of those antibodies. The only question is, how long is it going to take? And we've just completed analysis of data from our study, from Finnish study, and from German study. And the good news is that not everybody develops diabetes in the next 15 years. But the bad news is that about 80% of kids who are positive for 2 out of 4 autoantibodies will develop diabetes in the next 10 years. So there's always a hope that it will take very long, but the younger the age of the kid at the time of detection of those antibodies, the faster it goes. Now, up to now, we have 74 kids in DAISY who have developed diabetes. And we've learned a tremendous amount of information from those kids, as well as from those 183. One piece of information that was kind of a shocker to not only parents, but also diabetes research community was that it's not an on-off switch. We kind of suspected, clinicians, that when we see a sick kid, the process probably started maybe 2 weeks ago, maybe 2 months ago. But when you look at the DAISY data, and we are looking at the glycosylated hemoglobin, A1C, which is a very good marker of average blood sugars, this marker starts going up still in the normal range up to 2 years prior to diagnosis. So it's a very slowly progressing process. I've had a number of difficult conversations with families coming from DAISY or TEDdy where parents just don't believe that it's happening. And we would like not to believe too, but when you see enough of those high blood sugars and A1C is already in the diabetic zone, I don't think there is any reason to wait any longer, to wait with insulin. So I hope it will never happen to your family. But if it does, trust us, Dr. Klinkenschmidt, Dr. Steg, and myself, then when it's time to start insulin, it's time to start insulin. And if you wait too long, your kid may get really sick, end up in emergency room, even in intensive care unit. And even if it does not get that bad, there is a good deal of evidence that if you are waiting too long to start insulin, more of those insulin cells will be killed, and there will be less endowment left for the rest of the life, or at least for the next few years, and diabetes will be a bit more difficult to control. Okay? So this is a very complicated slide, but sends just one message. It's both genetic and environmental. People ask me, is type 1 diabetes just genetic? No, it's both. You need both pieces to cause this inflammation in the pancreas. And we've learned over the past 15 years or so that there are multiple genes for type 1 diabetes. The strongest genes, susceptibility genes, are in the HLA region. And those are the genes that we use in screening that Judy mentioned, TEDDY screening, DAISY screening. We use those gene markers to pull maybe 5 or 10% of kids from the general population who are really at increased risk. But by saying those are at higher risk than the others, we are leaving 40% of future cases of diabetes in this kind of low risk pool. So as a newborn screening, it would be abysmal. No, it would not pass any review board. It's just for research purposes to narrow down the number of families that we bother to participate in those studies. We are going for the highest risk families. And in terms of the environmental part, we've learned that many of those factors seem to be involved, and there have been clinical trials to eliminate cereal, baby diet, to eliminate cow's milk in early childhood, the trigger study, and even to add vitamin D or omega-3 fatty acids. You'll see some data later from DAISY. Unfortunately, none of those studies have shown any benefit so far. So, excuse me. I believe personally that there's a big factor that we still don't know about that probably interacts with the HLA system somehow. And this is the reason that we are continuing those studies to find the true answer to the etiology of type 1 diabetes. Now, this is a very, very busy slide. Probably the most difficult slide that I'm going to show today. But I would like to spend a little bit of time on explaining what we are really showing here. So, how many of you have bought a car in the past 5 years? Raise your hand. Okay? And do you know the true value of your car? At that time, did you know? No. I know it changes, it goes down, but on the day that you signed the papers, did you know the true value of the car? Nobody knows, right? Because there are so many factors. Of course, you could do research online. You could ask your friends. You could negotiate with the dealer. You are looking for promotions and all of that stuff. But at the end of the day, you are never sure if you got the best deal or if it's the true value of the car. You have a range, right? You know that you shouldn't pay more, and this is kind of illustration of this. You shouldn't pay more than certain price. This is the price that you paid, right? Let's say $20,000, and you know that you shouldn't pay more than $25,000 for this car, and you know that on the best day you could get it for maybe $18,000, right? So this is the same situation we are dealing when we are trying to answer the question if a gene or cow's milk or any other factor it increases the risk of diabetes. We know only that the true risk is in this range, and using the data that we analyzed today, we got an estimate somewhere here, but in a different sample from a different population, the estimate may be here or there. Okay, so this is our confidence region. But when you look at this pattern, different genes, even within the HLA region, can make the diabetes more likely or can prevent diabetes from happening. So there's a range of genetic effect. And by the way, blue bars show the risk of developing inflammation in the pancreas, while the red bars show the risk of progressing from inflammation to diabetes, to high blood sugars. Okay? And those are very consistent. So those genes regulate both steps, initiation of the process and progression to diabetes. So here's about 20 papers out of the list of 100 that you got. We are not going to spend more time on genes today because we cannot change those genes, right? We cannot do gene therapy or anything like that. So we just use this information to improve our prediction. When we talk to the next family and have this information, we can tell them a bit more precisely what the risk may be. Now, those are the environmental factors that we are looking at with the hope that we can actually modify them, avoid them, or if they are protective, put them in diet or in some kind of a vaccine. So, again, busy slide, but a few bars. We looked at enteroviruses. Those are the viruses that I mentioned earlier on. As a young fellow, I saw the epidemic of enteroviruses, and it was not completely off. A lot of people look at those viruses over the years. And in DAISY, very early on, we published an observation that was contrary to the common belief and contrary to my earlier beliefs, that infection with enteroviruses, as we could detect it in stool, serum, or saliva, was not a risk factor for development of islet autoantibodies. There was a very wide confidence interval, so we weren't quite sure, but if anything, it was on the protective side of this distribution. Well, at the same time, our Finnish colleagues were reporting something very different. They were reporting that in their population, presence of enteroviruses in serum or stool was very strongly predictive of development of those antibodies. And we argued at different meetings, and then NIH decided, well, stop arguing, just start working together. And this is how TEDdy happened. It was good time. There was surplus in the federal government budget, and the Juvenile Diabetes Research Foundation convinced the Congress to set aside $100 and then $150 million to find the cure and prevention for type 1 diabetes. And this is how TEDdy started. And this is still how TEDdy is possible in this difficult fiscal environment. Due to people like you or your family members who have been affected by type 1 diabetes but joined the effort with JDRF or just writing letters to your congresswoman or congressman asking to renew this program, to finish the job. And over time, more studies from Finland, from Norway, from Australia were published, and the picture is still unclear, but something emerges that seems to be a consensus, that when you see a virus in serum, especially in kids who already have islet autoantibodies, it seems to be a strong predictor of progression to diabetes. We found it in DAISY, and our Finnish colleagues found it in the DIP study. And now TEDdy is ready to give the final answer in a definitive study that I will show you in one of my last slides. Similar kind of pattern for dietary factors, and I'm sure a lot of you are interested especially in diet because this is what you can control, or at least you think you can control, right? You cannot control viruses that well, but you can add vitamin D or subtract gluten from your child's diet. So pay attention. This is what we found, and it's in the context of other studies that looked at the same factors. first cow's milk. There was a lot of talk about the bad influence of early introduction of cow's milk formulas on the risk of type 1 diabetes. It started about 20 years ago, and about 10 years ago, a large clinical trial called TRIGGER was initiated. This trial will show their kind of preliminary results next year. Right now, we don't know what it's going to be. But from the observational studies here in Finland and in Germany, it looks like there shouldn't be any effect of elimination of cow's milk on the risk of antibodies. We don't know much about diabetes yet, but on autoantibodies, there shouldn't be any risk. Now, these two studies, ours and the German study, Baby Diet, found that if you introduce cereals, doesn't matter, gluten-containing or non-gluten-containing like rice, too early or too late, the risk of autoantibodies goes up. Why? Nobody knows. Is this truly the cereal in diet, or is this just a marker of something else that is happening to your child? But when people call me asking, oh, no, I'm going to have a new kid, we are going to have a new kid, what have you learned from Daisy or Teddy? What should we do differently with this kid so she doesn't get the diabetes? I tell them, talk to your pediatrician and follow what he says. Because what the American Academy of Pediatrics says is to introduce all kinds of foods in small amounts between ages 4 and 6. This is the best time when, and especially if mom is still breastfeeding, this is the best environment to introduce new foods. They are better tolerated by the immune system, and then you can increase the daily amount of cereal or milk without seeing adverse effects. It's true also for celiac disease, it's true for asthma, it's true for a lot of allergies. So there seems to be a good period of time in child's development to introduce those foods. Some people say, well, can I exclusively breastfeed until the age of 1 year or even 2 years? And we have some moms, mostly from Boulder, who try to do this. Every year we have a couple of such moms. I personally have a couple of such moms in the clinic with a child who was newly diagnosed with diabetes. So I don't know for sure if it gives a little bit of prevention, but I know for sure that it does not give complete protection. Sorry about that. Omega-3 fatty acids seem to be protective, but only if you give them during the first year of life, and only protective from developing of those autoantibodies early in life, like during the first 2 or 3 years of life. The effect of those supplements in the first year of life seem to wane later on in life. Vitamin D, it would be wonderful, but it doesn't seem to have an effect on the risk of antibodies. I know I'm getting grossly behind time, right? So very quickly, this is what we are dreaming of. We want to prevent autoantibodies. We want to prevent the first step. And I strongly believe that if we find what causes this first step, appearance of autoantibodies, we will be able to control the rest of the process. If we are starting to prevent diabetes here at the late process when there is not much of insulin cells left in the pancreas, it's late in the game and it's very hard to change the course. So here are some of those trials that I already mentioned. One with called NIP, with supplementation with 3 fatty omega fatty acids. It didn't influence the rate of autoantibodies. Trigger, we still don't know what the results are going to be. delay in gluten exposure, no effect on the risk of diabetes, and a few more trials that were also negative. Now, there's so much talk about type 2 diabetes, obesity, and a lot of people say, well, maybe it's too much food. Maybe it's just obesity that is driving this epidemic of type 1 diabetes as well. So we looked in DAISY very carefully if BMI, body mass index, or obesity weight predict development of diabetes or inflammation in the pancreas. And we found something very interesting. Specifically, weight gain or BMI had nothing to do with development of autoantibodies or progression to diabetes, but the height growth velocity, higher height growth velocity did predict both steps. So, am I saying that you should ask endocrinologists, if you happen to have a tall, skinny kid, you should go to endocrinologist and ask, stop growth, please. No, I don't know yet what it is. It could be that we are seeing the effect of tall Scandinavian genes in our population, and those Scandinavian families tend to have taller kids and tend to have higher risk of diabetes. I don't know what it is, but it's definitely worth following. How to control? But if your kid is a little bit on the heavy side, don't agonize about it. Don't go into food fights. His risk of type 1 diabetes is probably as the same as for the general population. Okay, I'll skip this one. And just to finish DAISY, we are seeing that kids can develop inflammation in the pancreas not only in early childhood up to the age of 7, but also, amazingly, some kids who are negative for 10, 12 years develop autoantibodies, regardless if they have somebody with diabetes in their family or not, also around the age of 10, 11. So this was a big surprise. We thought all of the action was here, and then it was just a matter of time, how long it's going to take to go on to diabetes. But now we see that teenagers and perhaps also older people may develop inflammation in their pancreas from scratch. Those patients don't progress to diabetes as quickly as those found to have antibodies earlier. They amazingly have the same genes that we know in terms of diabetes genes. They tend to be a little bit more coming from minority groups, and usually the first antibody is GAT, not insulin autoantibody. So there's a growing amount of evidence pointing that type 1 diabetes the process leading to type 1 diabetes may start at any time during the lifespan. It may start mostly it starts in young kids, but it may start in teenagers, young people. My oldest patient with type 1 diabetes that I didn't really treat for a long time was diagnosed at the age of 80, 81 to be exact. So half of the patients diagnosed with type 1 diabetes in this country are older than 20 at the time of diagnosis. So one half. We use this argument a lot when we ask NIH to continue funding, because we tell them, look, we are not done yet. We've figured out what's happening in early childhood, but now we have to figure out what's happening during puberty. And some of your kids in DAISY may be already beyond puberty, maybe already entering colleges. We still want to keep in touch with those kids and at least see if they are okay, if they don't have antibodies or diabetes. All right, TEDDY will be easier. It's a bigger study. Judy told you a lot numbers already. As of last June, TEDdy have identified already 425 kids with persistent autoantibodies and 117 with diabetes. So you see that we'll get answers, better answers quicker in this collaborative study with bigger numbers. Now, so far, we've published 21 papers, and you have the list, and you have some abstracts. But honestly, most of those papers just describe the protocol, procedures. Suzanne is going to tell you some exciting findings from the psychosocial community committee that have been published, and I'm not going to talk about that. I'd like to just bring up one paper of clinical importance that came out from TEDdy a couple of years ago. We looked at the frequency of diabetic ketoacidosis, a very dangerous complication following high blood sugars that in this country affects about 40% of kids newly diagnosed with diabetes. Now, remember, 40% if you are not participating in our studies. And it's not a commercial. If you participate in our studies, it's only about 13 to 15%. It's about 15% in kids who are younger than 2, and about 15% in kids who are younger than 5. So in America, we beat hands down the routine clinical care in terms of preventing diabetic ketoacidosis. Countries like Finland and Sweden have slightly better healthcare systems, and the rates of DKA are lower in TEDdy and DAISY, but not significantly lower for kids older than 2 years. So fortunately, you live in this country, so I can take the credit on behalf of the studies that by participating in those studies, you have significantly lowered the odds that your kid will have to go to the hospital when diabetes happens. And this is consistent with earlier data from DAISY. In DAISY, in addition, we found that the dose of insulin in those kids who were diagnosed by DAISY compared to kids from the general population, the dose during the first year of treatment was lower. So those kids had more of their own insulin cells preserved for at least a year. And now we are trying to follow up on this information in this study funded by the JDRF called Post-Diagnosis Study in TEDdy. So if, God forbid, your kid is diagnosed with diabetes, we will approach you asking to participate for 1 or 2 more years in the follow-up to see how much insulin cells were left despite progression to diabetes. Okay? Now, what TEDdy is going to do in the next 2 or 3 years? We are well funded, so we'll continue following the cohort. We want to keep all of you in the intensive follow-up. If you have to move somewhere, let us know how we can make it happen. We have ingenious staff in terms of doing this long distance and at a lower burden to you. But we really need to keep everybody in the study as much as possible to meet those goals. We are already using the samples, blood samples, stool samples, nasal swabs, not yet, but the saliva samples we are using in laboratories to specifically look at the following potential risk factors. In terms of dietary risk factors, we are going to run about 13,000 samples in a lab in Finland to measure in each of those samples content of fatty acids in red blood cell membrane, which is the best marker of kind of saturation and exposure to good levels of omega-3 fatty acids, and those vitamins as well for each of those vitamins, there have been publications showing risk or benefit. So we are going to follow on those leads. We are going to see what genes out of about 20,000 genes that your kid has, which genes were expressed at several time points before development of autoantibodies. So we want to see which pathways lit up, were turned on or suppressed. And this is a human, I cannot even describe the amount of data that this will generate, but think 20,000 genes times at least 8 time points, and 400 kids who developed antibodies, and about 1,200 kids who have not developed antibodies. So it's a tremendous amount of data that we will sort through and find those genes and pathways that seem to be activated or deactivated in the months and years leading to development of inflammation in the pancreas. And microbes. Teddy is teaming up with two world-leading institutes that do human microbiome work. What is microbiome? This is the stuff, bacteria, viruses that live inside us and on our skin, in our mouth, in our ears. So if you count all of those bacteria, the cells, there are more cells, bacterial cells, inside us than human cells. And we carry about 28 pounds of that stuff inside or on us. Isn't that amazing? Yeah, 14 kilograms. So somebody finally discovered, whoa, maybe this stuff has something to do with many of the diseases that we are suffering from, like obesity, like heart disease, type 2 diabetes, type 1 diabetes. So now we could talk about this for 2 days, but basically what we are going to do, we are going to find out exactly what bugs your kids carries, and what families and species those bugs belong to, both bacteria and viruses. And we are going to do it in all the stool samples and blood samples that we received from you. Are we going to find the single agent that causes type 1 diabetes? I hope. But most likely we'll see some perturbances, some waves or changes that may give us leads. For example, some of us believe that antibiotics are overused, and once you put a kid on antibiotic, this whole composition of bacteria in the gut changes. And so if you do it too often, this may be related to autoimmune diseases. But I guess this is a story for another day. I will just end with this increasingly complicated picture, just to impress you. We are also going to look at about 800 small molecules that we can find in blood. And those are stuff like amino acids, fatty acids, little products or metabolites of carbohydrates, protein, or fat, and also stuff that comes from those bacteria inside us. So we are going to use what is called metabolomics. And simply speaking, the blood sample is put in a big machine, and then a lot of magic happens, and you get a picture like this, where each of the small molecules that was found has a peak, and this peak can be compared to kind of a standard. So we know if the amount of that metabolite is higher or lower than what we see in the control group. And then there's a lot of high-level statistics. We actually have done it for DAISY already. And there are some fascinating findings, but I cannot tell you what they are because we have to publish this paper first. So if you really want to know, come back to the next session. And I will end with this, and hopefully, the way to preventing type 1 diabetes turns out to be more difficult than we thought. But I hope we are not somewhere here. I hope we are already getting out from the clouds and seeing the goal. Okay. Thank you very much.
Well, I'm gonna. That was a lot of information. So, do you have any questions for Dr. Revers? We hold all the questions to the end? Okay, I just want to make sure there wasn't a burning question. Whew, a lot to absorb, huh? Here's a little break. So when you're not here, it's very quiet back in that clinic. This is the lab. This is the playroom. Look how neat it is. And this is the clinic hall. It's quiet. The lights go off. If nobody walks down that hall, the lights go off. This is our office hall. It's just so quiet. And then it all comes alive, thanks to you. And kids are there. The playroom's getting activity. And we have happy staff who are working hard in the lab and on their computers. And so this is what goes on a little bit behind the scenes, as well as in the front. Here's a few facts. So I was talking about DAISY screened 33,000. Teddy, Colorado screened 76,000. DAISY, it took them 10 years, and they did it in one hospital in St. Joe's, with an average staff over that 10 years of about 3 people working on it. Teddy got to that almost 77,000 over in 5.5 years in 12 hospitals. The way we could do that was 12 hospitals with an average staff size of 20. Part-timers, full-timers, people working. Our goals on both studies were to cover those hospitals as close to 365 days a year as we possibly could in order to meet the goals, the enrollment goals that we had for the studies. So, important difference. A few more facts. The youngest in DAISY is 8, but there's a few people who have been enrolled after sort of post the closure of the official enrollment. So that's what the 3 is. Our youngest in Teddy is 2.5. The oldest is 24.5, 25, almost 25 years old in DAISY, our participants, the kids that aren't kids anymore. And our oldest in Teddy is 8 years old, just turned 8 years old. September 2004 was when we started screening. So those are our first kiddos. Median age is 14 in DAISY, 4.5 in Teddy. That's the age at which half the cohort is above or below that age. So the greatest number of visits, we were trying to think of what might be interesting. And we learned a lot trying to do this. So in DAISY, the highest number of visits by one child is 46 visits. Isn't that impressive? And in Teddy, remember, Teddy's only been going 9.5 years, 29 visits. It's a bit more intensive protocol. So 29 visits I was just sort of stunned by that. So I just look at all the work that's just symbolic of all of the incredible amount of dedication on the part of our families. And this is just to highlight that. And where does your data go? Well, it gets entered into the computer by hardworking people. We scan it. I should have taken a picture of the scanner for Teddy. DAISY stores all of your samples, all of that stuff down in freezers. We got a freezer farm, we call it, down in the basement of the Barbara Davis Center. And freezers labeled Daisy like that. Teddy sends theirs off to the NIH repository. Because all of the Teddy sites are sending it into the repository from Finland and Sweden and Germany, plus all the 3 sites, Georgia, Florida, Colorado, and Washington, are sending to the same repository. When they had they had the storm and the outage, I was a little worried and was really hoping that, because it was a week. Washington was down for a week. And so we were lucky, we didn't lose anything. We scan our data to our data coordinating center, but we have to keep your paper files. Remember, 29 visits for Teddy, 49 visits for that Daisy. We have to keep all of that. And so we have a file room, but then everything has to go to Iron Mountain, has to be stored and secure. I know this doesn't look very secure, but it's behind a locked door. And when we need to get to it, this is the staging area, but we're overrun with paper and data. So, but we have to keep it organized and be able to get our hands on it, because if somebody comes and does an audit, we have to be able to find it. So this is what we do with it. So I'd now like to turn, with that little moment of levity, I would like to now turn it over to Dr. Suzanne Johnson, my esteemed colleague and friend. All right. Who do you want to?
So I was in, can you hear me? Maybe you don't want to hear me.
Hold on. We'll get it right. There we go. Okay.
So I had the opportunity to come to Denver to actually give a talk somewhere else, and so I told Judy about it, and so she decided I should give another talk. So what I want to talk with you tonight a little bit about is, so what does psychology have to do with being risk for type 1 diabetes? People find it kind of surprising that psychology has anything to do with these studies. Oops, she told me to use the left one here. So let's start out, well, what is psychology? You probably all have some ideas about what psychology is. Well, psychology is actually a science, and it's a science about what we think, what we feel, and what we do. And it's also, clinical psychology is also a health profession in which we apply psychological science to improve people's lives. So essentially, I'm a clinical psychologist. I'm a clinical child psychologist. And I'm really just like Dr. Rivers. Dr. Rivers, though, he's a physician who studies medicine, who does science, the biology of disease, and he applies that science to improve people's lives. So I do the same thing, but I'm interested in thinking, feeling, and behavior. So how did I get involved in all these studies that you're learning about? Well, I was a psychologist who worked with type 1 diabetes patients, just like Dr. Rivers. And I always worked on what we call interdisciplinary teams. So I saw my patients with physicians, diabetologists, and I also did research just like Dr. Rivers. Well, then we, in diabetes, came up with this way of doing these biologic assays that indicated that a child was at risk for the disease before the disease had even occurred. This caused a lot of controversy because there were many people who said we should not be doing these tests on kids and finding out who's at risk when we have no way to prevent the disease. And they argued that the reason we shouldn't do this is because it would cause a lot of psychological problems. So my colleagues, since I'm a psychologist and I work with kids with diabetes, asked me, would you please address this issue? What kind of psychological impact happens when you screen kids for a disease, find out who's at risk when you have no way to prevent it? So that was my job. So I'm going to talk to you a little bit about what we've learned so far. So we're going to start out with thinking. What do you know about your child's diabetes? We're very interested in the concept of what we call risk perception. Do parents accurately understand their child's risk? So this is what we've learned so far. As Dr. Rivers has already told you, and you already know, type 1 diabetes risk is what we call a probability. It's not a guarantee. He just showed you a million slides. He's been working on this for 30 years. Just because your child's at risk is no guarantee your child will actually get the disease. But probability is really difficult for people to understand, and it's really difficult to communicate it effectively. So what we're learning are better ways to talk to families about risk. And this work actually is informing the larger science of the human genome, because actually most diseases are like type 1. Most diseases, if you have a gene, it isn't a yes or no. If you have the gene, you get it or you don't get it. There's a few diseases like that. But most diseases are like type 1 diabetes. You have a risk for getting the disease, but it doesn't guarantee you will get it. And we suspect that most diseases are a product of these environmental gene interactions that we're trying to understand in type 1 diabetes. So what you're helping us to do is do something very important about type 1 diabetes, but you're helping us do something even bigger than that. Because it's very similar to a lot of diseases that we're trying to understand. So what about feeling? What have we learned about feeling? So our focus has really been on how worried or anxious parents get when they learn their child's at risk. So this is what we know so far. First of all, being mildly worried or anxious is very common, completely normal. This is not going to surprise you. Moms worry more than dads. And if you are a parent where you already have type 1 diabetes in the family, either you have it, your spouse has it, or another child has it, you tend to worry more. But we also know that with time, anxiety and worry declines. So what about behavior? What have we learned about behavior? There's a couple of things we're very interested in the behavioral area. The first is what we call monitoring. That's where you're looking for the kids to get it. So doing things to see maybe they're getting diabetes. And also prevention. Even though we have no known means of preventing the disease, Dr. Rivers just discussed that, we're still very interested in whether parents try to do something to try to prevent it on their own. This is what we've learned. So monitoring prevention efforts are common. Lots of people that do them, but not everyone does. Parents, if there's already type 1 diabetes in your family, you're more likely to both monitor and try to do something. And what we found, if you monitor or if you try to do something, generally what people do is not very harmful. So in terms of behavior, the other thing that we're really interested in is who joins studies. What is it about you that you join Teddy or Daisy? Who joins? Who stays in the study and who drops out? Why people stay in the study? And how we can improve the study experience so that people are more likely to stay. So who joins? So this is not going to surprise you either. Again, if there's type 1 diabetes already in your family, you're more likely to join. If you're a parent from an ethnic minority group, you're less likely to respond to communications that we send you about joining the study. Interestingly, if I actually get to talk to you, you're equally likely to join the study. But I have more difficulty getting to talk to you to begin with. Older moms are more likely to join the study. We can discuss what it is about us older moms, but the primary reasons, this is not going to surprise you, that people decline are the protocol's too demanding, especially blood draws. This is a major concern for families, and just people are very, very busy. So who stays in Teddy and who leaves? Again, if you already have type 1 diabetes in your family, you're more likely to stay in the study. If you're an older mom, you're more likely to stay in the study. If you have accurate risk perceptions, remember I talked to you about thinking, if you are accurate about your child's risk, you're more likely to stay in the study. If the dad participates, even in some small way, the family is more likely to stay. These are two interesting findings. If you work throughout your whole pregnancy, you're more likely to stay in Teddy. And I think that's interesting. We have all kinds of speculations about that. And if you smoke during your pregnancy, you're less likely to stay. So, one theory is if you smoke, perhaps you're not as health focused, and so you might not be as committed to some study like Teddy. Our speculation is that moms who manage to work through all of their pregnancies are really good copers, so they can manage Teddy as well. So why do people stay in Teddy? And these are what you all tell us. First, number one, they stay because it makes them feel comfortable that someone is watching for diabetes. And Dr. Rivers actually, the study that showed that if you're in Teddy, you're less likely to ever have DKA, is very consistent with that, with this concept that actually watching for type 1 diabetes is a good thing. But people give other reasons that I think are really important. One is that they want to help science discover the cause. They also want to get their kids' antibody results. They want to sort of know, is there any evidence if the child's progressing? Another reason is the relationship with the staff. Many families get quite close to the people that they're seeing in clinic. And the last reason, big reason that people give is the possibility that a child might be able to participate in a prevention trial that might become available. So what have we learned about improving study retention? We actually have become quite successful in identifying people that are likely to leave the study. We call them high risk for leaving the study. And we just did an interesting project where we developed a tailored approach to these families so that their experience was more positive. And we were able to show that these families were more likely to remain in Teddy. So getting back to the question, at risk for type 1 diabetes, what does psychology have to do with it? Well, as I mentioned, psychology is the science of thinking, feeling, and behavior. And learning that you or a loved one is at risk for type 1 diabetes does have psychological impact. And understanding that impact is important not only to studies of children at risk for type 1 diabetes, but for all of medicine, as we seek to identify and treat at-risk people early before they develop a disease. So recruiting and retaining children at risk for type 1 diabetes is absolutely essential if we are able to figure out what the triggers of this disease are. And these are very valuable participants critical to our ultimate ability to develop a prevention for these diseases. Recruitment and retention are behaviors. They're psychological phenomenon. Psychological expertise in the study team enhances the likelihood of a positive participation experience and study success. So, love, curiosity, relationships, goodwill, appreciation are powerful psychological phenomena. Maybe you're here because you love your child and you want to do what's best for him or her. Maybe you're here because you're curious and you want to know more. Perhaps you're here because of the relationships you've developed with the study staff. And perhaps you are here because you want to contribute to science and make the world a better place. I just want you to know that we appreciate you, and we hope you stay in Daisy and Teddy. And you're central to making type 1 diabetes prevention a reality. We simply cannot do it without you. Thank you very much.
Now we have time for some questions, and we're encouraging our other investigators who are here, Dr. Georgiana Klingensmith and Dr. Ed Liu. Dr. Liu is a gastroenterologist, pediatric gastroenterologist, and Dr. Klingensmith is one of our premier clinicians and researchers at the Barbara Davis Center. So, and they're all investigators, so you can ask them any kind of question you'd like. What do you think? What do you got out there? Let's get some mics going. Who's got a question? Great. Hold on, I gotta get things turned off here.
I'm from Boulder, and I have a breastfeeding question.
And she has a child in both studies.
You mentioned that some moms will exclusively breastfeed for 12 months, and there's no benefit or risk associated with that. What about extending the duration of breastfeeding, like say beyond a year? Does that, when the child is also eating regular food, does that have any protective effect at all? Yep.
Starting is not pregnant and breast milk already around the age of 6 months. So exclusive breastfeeding may be a mistake because the breast milk is a fairly rich nutrient, but is lacking some vitamins and micronutrients. So it's very good infant up to the age of 6 months, especially in countries where infections and kids are still around, like Africa, Latin America, because it protects the child from unattended infections. So it's also clean and safe in terms of ability to prepare for it. But in our society, I think there is a good intent around the age of 6 months should not be the exclusive for breastfeeding. And I think then as we look at age, extend maybe
My question isn't really about exclusive breastfeeding. It's if there's any benefit to continuing breastfeeding when the child is having normal diet, say up to a year of age. Thank you both.
Around the time of delivery. I can speak louder if.
There you go.
There. Okay. It's just a little sort of flaky.
Uh,
There.
Thank you. So,
Sorry.
but it's basically. I will stop here. Okay.
Okay. Okay. Next question.
Yes, I had a question. In the study, you also test for celiac antibodies. And I'm wondering if you have any data or there are any studies, probability studies, that show the likelihood of progressing from celiac antibodies to diabetes, type 1 diabetes antibodies, and then on to type 1 diabetes.
I'm sorry, can you repeat that question? Just in terms, were you asking about the association of celiac disease and type 1 diabetes, or just the celiac disease
right,
itself?
Are there studies like you showed that show the probability, the likelihood of progressing from celiac to type 1 diabetes?
Okay, so the other way around, because right now there are recommendations that if someone has type 1 diabetes, that they should be screened, regularly for the development of celiac disease. And I've always wondered about that. And so for the kids that we take care of with celiac disease, right now with the GI societies, there's no recommendations at this point to screen them proactively for the development of diabetes. I think the reason for that is because the incidence of diabetes, if you have celiac disease, the incidence of diabetes is slightly higher, but it's not as high as if it were the other way around, if you had diabetes first. If you have diabetes, the risk overall for developing celiac disease is about 10%. If you have celiac disease first, when they look at this, the risk is maybe about 2.5-fold for developing type 1 diabetes. But I think in the future, if we start doing, the HLA testing for our kids, just because there's certain combinations of the HLA genes that put you at a higher risk for celiac disease, and certain HLA combinations that put you at a higher risk for type 1 diabetes, someday we might be able to actually personalize the screening so that if you have celiac disease and we know that your HLA puts you at a higher risk for diabetes, these may be the kids that we actually proactively screen for the development of diabetes. But right now, there's no recommendations for that.
Hi, I have several questions, and they're very scientific. I'm a researcher and an immunologist. And I went to a seminar recently from John Cambier at National Jewish, and he is looking at the way that anergic cells that are insulin specific become non-anergic, that they start binding insulin, and that it would be a precursor to the development of antibodies. And I wondered if this was something that you guys might be interested in researching as an earlier time point, and if you've already talked with him, or it might be a good idea to talk with him about that.
Well, it's a small city, and is it on? Yeah. And of course, we are working with John, and we have George Eisenbach, who is a famous immunologist on our team, and colleagues from all over the world who shares the same desire to look not only at antibodies, but also at T cells. T lymphocytes are the real menace in diabetes. They do the killing. They shoot those bullets that kill insulin cells. Antibodies are just a marker. It's kind of smoke that you can detect, but it's not bullets that are dangerous. So if we are ever to prevent diabetes or cure diabetes in those kids who already have it, We have to learn how to teach our immune system again to tolerate those insulin cells. The problem now with, let's say, islet cell transplantation is that when you put new islets in, and they are usually put in the liver, you are fighting two battles at the same time. One battle is rejection, like in any transplant, like in kidney or heart rejection. But the autoimmunity comes back, and there are specific lymphocytes, like you are saying, that come back just to hunt those insulin-making cells. So until we can learn how to control autoimmunity, the transplantation, however many we have insulin cells, is going to be very difficult. But those are excellent questions, and our team is looking at the crosstalk between T cells that are the effector cells and antigen-presenting cells that are the messengers. So antigen-presenting cells have those HLA molecules on their surface. They bind something that starts the signal, and the experiments that you are describing, it's a piece of insulin that starts a battle. But it's quite likely that there are other antigens coming from environment. For example, in celiac disease, it's not insulin, it's a piece of gluten that is presented by antigen-presenting cells in the context of the HLA to the T lymphocytes, and they do the damage.
Right, that actually leads me into my next question, because I work with chronic beryllium disease, and if you're familiar with Andrew Fontenot's work, it takes the HLA-DP2 plus a self-peptide plus the beryllium molecule, the environmental trigger, and so your big yellow circle, I mean, could it be possible that one of these, not insulin, but one of these other environmental determinants, some part of it, is interacting with with an insulin-specific peptide or something? I mean, that could tie some of this other stuff together. Is that the environmental trigger?
I think you're on the right track, and this is kind of a hapten situation where you have an environmental agent and also a protein or a piece of protein coming from our own body. They form a complex. Similar situation in celiac disease, where you have gluten and the transglutaminase. They form a complex, and that complex, not either of those parts separately, triggers the autoimmunity. Very good.
Do you think it's possible that the second thing that's happening on top of the genetic predisposition could be happening in utero?
Well, I think that what happens in utero may be important, and in some of in DAISY studies, we've looked at maternal infections during pregnancy, and actually minor infections and illnesses decreases the risk for later diabetes in the child. Children don't show evidence of autoimmunity until after 6 months of age. So we don't see any evidence of autoimmunity or antibodies during that period of time. So we can't rule that out, but we don't have any direct evidence of whether there is something, what it might be. We're still looking.
My second question is, kids who are diagnosed with diabetes, type 1 diabetes in adults, there's the association with depression. Have you guys found any, have you found anything about the psychological impact of the kids that are diagnosed when they're part of the TEDDY or DAISY study? Does that somehow lessen that psychological impact? Does it? Do they struggle less when they're teens?
Well, I'll try and answer this. So with TEDDY, the kids are very young so far, but we have, Dr. Rivers mentioned, we do have this new protocol for when the children, those children that get diabetes, they are going to go into this sort of post diagnosis study funded by the Juvenile Diabetes Research Foundation. And as part of that, we are going to be looking at how the family copes with it and how the child copes with it. Our hypothesis is that children that in families that have been in TEDdy may actually do better than families that where this just sort of comes out of the blue and they're surprised. But let me say one other thing. Having worked with kids with type 1 diabetes my entire professional career, I really think it's very important that you understand that children and families are very resilient. And although, yes, some children become depressed, people without diabetes become depressed. So that's sort of that's very important to me, that people don't pathologize type 1 diabetes or end up feeling that if your child gets type 1 diabetes that they're going to have a lot of psychological problems, because I don't think that's the case. Yes, there is quite a bit of research in adults looking at the higher prevalence of depression. There's a correlation between depression and diabetes. It's a very interesting phenomenon, quite common in type 2 actually. And most of that research has been done in type 2. We could talk about that, but it's very interesting. We don't know whether depression actually precedes it, the type 2 diabetes. You could have some hypotheses about that.
There's some work that's suggesting that the depression and psychiatric or psychological issues in adults with type 1 diabetes is more prevalent in those who have complications, which we hope we're going to prevent with modern therapy.
Let me maybe go back to your original question just for a second. The clinical observation, also anecdotal, from the group of 74 kids with diabetes. I follow maybe 10 or 15 myself. Georgiana probably has similar numbers or larger. I don't see any difference in terms of negatives in kids who came out from DAISY or TEDdy. They pretty much represent the cross-section of the society in terms of, some and many are in excellent diabetes control. Some are in the middle, and there are some kids who just don't care, and families that don't care that much about blood sugar control. But if just eyeballing, I would think that those kids do on average better in terms of A1C and kind of organize this structured way to approach their diabetes, although there are exceptions. But I don't see a lot of depression in those families.
Hey, good night. First, I will try to put all my words in order because I'm originally from Colombia and English is not my first language. So I have a question. I have a question. Only kids that has the gene can develop diabetes, or maybe there are other kind of environment factors that can, in the future, can help, can develop diabetes. And other question that is linked with that is because I know that this study only have kids that had the genes, and you are studying the environmental factors and the gene factors. So the question here is, if there are kids that develop diabetes without had the genes, maybe there are common factors between TEDdy Studio or DAISY Studio with these kids that develop diabetes and didn't have any kind of genes.
It's a very interesting question because, and a lot of people are asking, look, those kids who have a lot of high-risk genes, I'm sure they are genetically predisposed, and a little bit of environmental influence will make them diabetic. What about those kids who have all normal genes, if you will, what makes them diabetic? They must have gotten a massive dose of this environmental agent to make them diabetic. And we don't know, but what is happening all over the world over the past 50 years is that when you look at kids who are diagnosed with diabetes, more and more of those, in terms of proportion, have lower risk genes. So the diabetes is spreading to kids who don't have the highest risk genes, which may suggest that the penetrance of force of those environmental factors that we are looking for is increasing. The data from Australia, Finland, England, and Colorado, now all different populations, shows the same phenomenon. I'm not sure if...
No, I agree with you. And of course, we're studying the high-risk gene pool, so we can't really comment too much on the fact that, the increase in the increase of diabetes seems to be related to, as Dr. Rever said, an increase in the number of children who don't have all of these high-risk genes. They may have fewer numbers of high-risk genes, and they may not have the highest risk genes. Why that is, we really don't understand. We would like to understand that. It doesn't seem, the obvious thing is the whole world is getting better nourished, shall we say, fluffier. But that doesn't seem, at least in the DAISY children, that doesn't seem to have contributed to it. So I don't think it's really that we're getting heavier as a world population. That's the only thing I can say.
Is there any indication that a gluten-free diet has an effect on the process of type 1 diabetes?
The short answer is no. There's just not enough evidence. I guess since I have the mic, I'm just going to go back to that very first question. Since it was our Boulder family who had, kids in both studies, in terms of celiac disease, the benefit of breastfeeding. I mean, there's no good evidence right now, but the American Academy of Pediatrics, they do recommend, exclusive breastfeeding for the first 6 months of life, and then the introduction of solids. It used to be between 4 months and 6 months, but just very, very recently has changed to 6 months of age. So exclusive breastfeeding and then the introduction of solids at 6 months of age. And then, they recommend breastfeeding up to the first year of life as long as it's sort of mutually beneficial for the child and the parents. I'm just going to step out on a little bit of a limb here, but, in Sweden, what they're recommending right now is the introduction of solid foods under, quote unquote, the protection of breastfeeding. And their hypothesis is that if you're being introduced gluten while you're under protection of breastfeeding, it may actually decrease the incidence of celiac disease. How this is going to sort of pan out with the AAP recommendations, we'll have to see, because the AAP right now is actually recommending that we're introducing solid foods at 6 months of age, around there, and really not that 4 to 6 month age period now. It's really recently changed, so we'll have to see how that works out.
We have someone with a mic in the back.
At what time period did the incidence of type 1 diabetes start to increase in American history? Was it like the 50s? When was that? That's my first question.
So there's very little data in terms of population-based numbers prior to 1965. And since 1965, the incidence has doubled every 20 years. So there are some anecdotal papers from the early 20th century which would suggest that this kind of pattern at least started around the Second World War. We don't know much really, mostly because there was a lot of other diseases at that time in kids. We don't remember that, but most of the kids were dying of diarrhea or pneumonia or tuberculosis. So this was just a drop in the bucket. Nobody paid attention to those few diabetic kids, except maybe for Boston and some better developed parts of the society. So, but data from Finland are the most solid, and the line is almost a straight line, starting from the early 50s. So if you extrapolate it back to, I don't know, to 100 years back, it would suggest that there were no diabetes at that time, but it's not true. There were people who were insulin dependent, looked like insulin dependent in the 19th century as well.
Right. Right.
But
so
the-
the steady,
the-
steady
the
incline
increase-
started like in the 60s, right around there is where we can trace it to at this point. Is that correct?
The increase in Finland, it looks like it started around 1985, 1990.
Okay.
That there was a pretty steady rise from when they have their earliest information.
Diabetes for the first 11 years of existence. So it looks like there are periods of peace and quiet, and then bang, it goes up. But it's not been going down since we've started this epidemic. So there may be some short-term triggers, but when you look more and more at those lines, it looks like there is a relentless increase of something in the society, or maybe continuous subtraction of something that we used to have. There's
right.
a hygiene hypothesis that pretty much says that it's good for kids to eat dirt, right?
That's right.
And hang out with dogs and cats and sheep and this type of stuff. This way, the body gets enough stimuli, infectious stimuli and bacterial stimuli, that the immune system is focused on what's coming from outside. It's really very simplistic, but no, this makes sense that in absence of those stimuli, the immune system may go array a little bit. I'm not sure if it's true, but going back to your question about pregnancy and type 1 diabetes, risk factors in pregnancy and type 1 diabetes, there's a really cool hypothesis called Herd hypothesis that postulates that women in reproductive age in our cleaner society are just not exposed to some of the bugs like enteroviruses. So when they become pregnant and happen to be infected, exposed to those viruses, they don't have natural immunity. So when they are infected in the first or second trimester, the baby actually gets this virus too, and the virus may establish chronic infection. So it will, the baby will, then child and adult will never get rid of this virus because it acquired the virus when he was in the womb and kind of immunoprotected. So this is one of the several theories that we are trying to follow on.
Well, I had another question still. So do you think that there's, and it's kind of two-part, and it goes along with what you were saying, do you think that there's any indication when you look at societal factors and dietary factors that processed foods and the use of overuse of antibiotics and antibacterial soaps and all of this stuff that we use? I know I have 5 kids, and my kids' schools promote the hand sanitizer all the time and all of this, and I've been very against that. And I would rather they go wash their hands with regular soap and water personally, just because I think, it's just almost an overuse where they're getting, and my kids, they don't get as sick as some of the kids who use that all the time. And I don't know whether that's just luck or not, but, do those factors into that in what you're looking at all or not?
So the difficulty here in terms of those environmental factors, like stuff that comes from burning gasoline or barbecuing or processing food, is that there are just too many of those factors to measure them. You would need liters and gallons of blood to really look at all of those, at each visit, I should say, from each child. So just to give you an example, when you look at food and how it's processed from harvest to King Super, on those veggies and potatoes, people have identified about 100,000 products, biochemicals, produced by fungi and other organisms that we don't even know they exist, bacteria, fungi. So 100,000 different stuff. And how can you tell which one is the one? So we kind of gave up on looking at all of those factors. We instead focus on the child. We look at what we can detect in the child, and not necessarily one little chemical, but we are looking at pathways. So if there is a pathway leading from one amino acid to another amino acid, or if there is a change in the profile of bacteria that we see in the gut, then this allows us to narrow down the search to maybe hundreds or at the most thousands of agents that may be plausible. Okay, so we are really very focused on the samples, on stuff that we can focus in the child rather than in the environment.
Do any of you know of any studies that are showing a correlation with infertility issues and subsequent treatment during pregnancy, such as metformin therapy for polycystic ovarian syndrome and the risk of developing type 1 diabetes later in life? And the second part is there an effect of the metformin therapy on the fetus?
To date, the American College of Obstetrics and Gynecology does not say that metformin needs to be discontinued, because metformin is used as a way to increase ovulation in some women. So that is looked upon as relatively safe during pregnancy for the fetus. I think that's really all I can say about that. We haven't looked at that specifically. We are looking at a study called the Today study, which is a treatment of type 2 in adolescents and young people, and following them. And we have had 700 plus young people in the study. We have had pregnancies. We are going then to prospectively look at the follow-up, where many of those women will be on not just metformin, but other medications to control their diabetes, to look specifically at outcomes there. I don't have that data yet.
Let me just add that there's a good deal of evidence that type 2 diabetes may have some of the origins in utero or early childhood. So to make it really brief, it looks like type 2 diabetes treated untreated in mom begets type 2 diabetes in the next generation. So this may be part of what we are seeing in terms of type 2 diabetes epidemic.
I had two questions that are totally unrelated, and I'll just give them both to you, and you can answer them in whatever fashion you want. The first was, all of your study appears, or both studies appear to be only with respect to developed countries, and I wondered if you had any information or data about third world countries and whether or not this rise is apparent in third world countries. I imagine it's more difficult to measure, and whether any anecdotal or other information could be gleaned from that with respect to environmental factors. The second question, I think, is more related to information you're likely to have that maybe Dr. Rivers alluded to, which is the microbiome issue, and I wondered whether there's any information coming out, whether probiotics might be helpful with respect to that.
So briefly, I came from Africa where I taught 250 doctors who are supposed to treat diabetes in many countries. And I did my best, but from what I gleaned from the data and from what people told me, 95% of kids with type 1 diabetes in Africa die either before they are diagnosed or shortly thereafter. And it's not necessarily lack of insulin, it's lack of knowledge. It's lack of providers and ability to teach parents how to take care of diabetes. So this is very sad, and it's to lesser extent affecting other developing countries. For example, my colleague from China several years ago told me that the child, among population of kids with diabetes, the longest surviving child had diabetes for 7 years. He could not remember anybody who would live more than 7 years after diagnosis. So those countries that we are kind of looking at and admiring because of their exports have still a lot of work to do in terms of catching up to the health standards of our country. We are very lucky to live in this country and have the resources that we have to take care of diabetes. So simple answer is there's no really research or knowledge about diabetes, not to mention clues from pre-diabetes. There are just no resources to study this. Your second question was more difficult because I'm not an expert in diet. I will take your name and ask Dr. Jill Norris, who was supposed to be here but couldn't make it, to maybe call you, if this would work for you.
On probiotics.
Yeah, sorry, I'll give you another short answer, but as far as I know, no evidence that probiotic, I mean, there's a couple diseases in GI that probiotics can actually help, but as far as we know in terms of preventing autoimmunity, preventing celiac disease, there's no role at this point.
Okay.
My question is in the TEDDY or the DAISY study or in a future study looking at children of mixed cultures and their genetic factors and their risk factors, since we're seeing more and more mixed marriages in multicultural children in our community?
In fact, we do collect the data in DAISY and TEDDY related to, in the U.S. sites, related to what people's ethnic and racial backgrounds are of the children, so that we can see the extent to which we can look at those factors as they relate. The European studies, interestingly enough, the sites actually don't look at their populations and classify them by these same definitions. So it's pretty much a U.S. phenomenon, and I would like to say that the Colorado site, TEDDY is done in 4 countries and 5 languages, and that's because the Colorado site is the only site of the TEDDY studies that it has the most number of Hispanic population, but it also includes people for whom Spanish is their first language, and we have a complete Spanish-speaking protocol that we started at the hospitals and when we were screening, and to ensure that we were actually capturing the breadth and diversity within that, which is our most common minority population. And so we have some potential to look at that here, and that will, for U.S. studies in general, that will be always a very important focus. Does that help answer your question? We have that data. We haven't looked at it yet because we haven't gone down that analysis path.
Maybe I'll add something from slightly different angle. We are looking at the rates of diabetes in different ethnic groups in this country. And what's fascinating, only 20 years ago, Hispanics and blacks had rates that were like 40%, at the most 50% of the non-Hispanic white population. Now, those rates are getting very close. So another evidence for influence of environmental factors that overcome Trump's genetics. As long as you live in the same country, eat the same food, go to the same schools, it seems that the risk is very similar.
Also, two questions. I guess one, especially the people here, I think would be good, but I'll go ahead and say both. Is there a way to find out about other potential studies? Because, obviously, especially these parents here, any information is awesome. And then the other question is there much of a connection between the different types of diabetes? Because, for instance, myself, I developed diabetes insipidus after my pregnancy, just shortly after. And from the genetic testing, I also have another type. So I'm looking at potentially 3 different types of diabetes with 3 different types of, ways to deal with them. I just didn't know how many kids or, hopefully not many have that kind of a thing. But.
Right. Diabetes insipidus has the name diabetes, which means increased urine output. Mm-hmm. But the insipidus means that mellitus means it's sweet urine. Insipidus means that it comes on of other causes and it's due to a pituitary or a renal problem. It started during pregnancy. It's usually due to a pituitary problem.
Okay, and so that wouldn't be anything that my daughter would be more at risk with?
No.
Because, and the other one is iron. So that would be doing the blood draws.
Right,
for iron.
Right. And that's more associated with type 2 diabetes, actually. Okay. So type 1 and type 2 diabetes really are very different pathophysiology. The causes of them, the genetics of them are quite different.
And so, now
Right, and type 2 is pretty common, so to have people in the family with both type 1 and type 2 is not uncommon.
Okay.
Oh, don't say that. Or we fall asleep.
My question was in regards to vaccinations. Since viral exposure can affect, developing type 1 diabetes, have you found any correlation with either risk or benefit of getting like the flu vaccine and whether it matters whether it's a live or dead vaccine? So I think the flu mist is a live vaccine versus the...
We didn't do the specific experiment analysis that you're asking for, but we looked at all of the routine vaccines, especially those given in the first year of life. If it's there, by chance, increase or decrease the risk of getting autoantibodies or diabetes. And to reassure all of you, all of the studies so far have been negative. So if you are wondering if you should... Right, no association. So if no direction, neither protective, no risky. So if you are wondering if, especially guys from Boulder, if to give your kid this next vaccine that he or she is due for, go ahead and do it.
And also, I think there was a study about the TB vaccine causing the rejuvenation of the pancreatic. Have you guys heard anything about that study?
it's really, we did years ago, in the early 1990s, a similar study giving children the BCG vaccine, which is the tuberculosis vaccine, and found no difference in rate of progression of loss of beta cell function with that. And in fact, the vaccine may have been worse. So we can't understand the Boston information.
I do.
Also, Finland used the BCG vaccine in the first 3 days of life. It was given to newborns for many, many years. And as you know, it did not protect kids from diabetes. All of the kids in Finland got this vaccine in the first few days of life.
Do your studies show any gender influence? And also, will the slides be available online anywhere?
Yes, the slides will be available online, either on the website or, and the Teddy Daisy website.
Just about gender influence?
Yeah, is there more incidence in type 1 diabetes in males versus females? Or?
Yeah, I mean, this is very interesting. When you look at our population, not much difference. When you go to highest risk populations like Finland and Sweden, more boys are affected. If you go to lower risk populations like Hispanics and let's say Asian Americans or Chinese, females are more frequently affected.
So one of the first charts showed the different countries and the prevalence of type 1 diabetes. And I know that some of the studies, there's been some studies to show that might indicate that vitamin D or sunlight had a part in that. How much of that the different prevalence in different countries is related to genetics? Like if you take the genetic risk factors of populations in those countries, and can you kind of normalize those curves so that to see if they're how much is genetics and how much is something else?
There's no final answer to your question because we still don't know the germ population frequencies of some of the newer genes for diabetes in some of the populations, but we know pretty much enough for the HLA genes, and the population frequencies of those genes do not explain the geographic difference. For example, in Finland, the germ population has as many people with high-risk genes as here. So it does explain differences between Asian populations and kind of European ancestry populations, but not within the European ethnic groups.
Okay, there are some relation between the average age that the parents are having the kids right now that it was before, because when I saw the graphic about how the diabetes increased in different countries, looks me like it's the same graphic that right now the parents we are having the kids. I'm more here in Colorado that the average is like in the 40s or the high 30s that we are having the kids.
That's a really interesting question. I haven't actually heard that discussed about age of parents and risk for type 1 diabetes. Do you?
Yeah, there's a fairly consistent, although not very strong, effect of older maternal age. Older moms, babies from born at older ages are slightly it's slightly higher risk than those born to young moms. Like 35 plus, yeah.